Well, good afternoon, everybody. My name is Kostas Panos. I'm a professor of electrical engineering and computer sciences at UC Berkeley and director of CITRIS and the Banato Institute. And today it is my pleasure to welcome you all to our seminar on behalf of the C3.ai Digital Transformation Institute. So our institute is relatively young, started this spring, and our mission is to attract the world's leading scientists to join in a coordinated and innovative effort to advance the digital transformation of business, government, and society. And it's a coalition of several institutions, as you see here. C3.ai is the corporation that is providing most of the funding in collaboration with Microsoft. We have University of Illinois at Urbana-Champaign, University of California at Berkeley, Carnegie Mellon University, MIT, Princeton, the University of Chicago, Stanford University, Berkeley Lab, and the National Center for Supercomputing Applications that are participating. But you have participants from beyond this family of institutions as well. This series of presentations, we started it early this summer, and we meet every Thursday at 1 o'clock Pacific time. And you can see here the exciting schedule of presentations that we have for next week and beyond. And you will note that many, if not all of them, are related to COVID-19. And this is not a coincidence. As I mentioned before in these introductions, we have started the Institute by leading a successful call for proposals on COVID-19. And many of the presentations, including the one that you see today, are related to the proposals that were being funded and they're executing right now as very exciting projects. The format of the presentation is pretty straightforward. We have a 40-minute presentation by a speaker. Hopefully we'll get 50 minutes of Q&A. You will note that in this webinar version of Zoom, there is the Q&A feature that you can use to ask questions in writing. And of course, if you see a question that you like, you don't have to repeat it, you can upvote it. And of course, we'll try to answer as many questions as the time will allow. So today's presentation has the title, Reconstructing SARS-CoV-2 Response Pathways, and is going to be given by Ziv Bar Joseph. He's the Four Systems Professor of Computational Biology and Machine Learning at Carnegie Mellon University, which happens to be my alma mater. His work focuses on the development of machine learning methods for the analysis, modeling, and visualization of time series high-throughput biological data. Dr. Bar-Joseph has been the recipient of the Overton Prize at an NSF Career Award and several conference-based paper awards. He's currently leading the Computational Tools Center for the National Institutes of Health Human Biomolecular Atlas Program. He has served on the advisory board of several national efforts, including the National Institute for Allergy and Infectious Diseases Systems Biology Program. Software tools developed by his group are widely used for the analysis of the genomics data. So very excited having him here to talk to us this afternoon. So I'm going to turn it over to him.
Great, Kostas. Thanks for the kind introduction. Let me share my screen. And thanks everybody for joining us. So as Kostas mentioned, I'm going to discuss some recent work we have done on trying to reconstruct pathways and cell responses to the SARS-CoV-2 virus, which is the virus that causes COVID-19. This is joint work. In fact, our project is a project that is funded by the C3.ai Institute, is a joint work with a group in Boston University, also a group at MIT. But what I'm going to talk about today is mainly a joint work with a group at Boston University. And the work itself was done by, by three talented postdocs in my group, Jose, June, and Ye. So if you look at the SARS-CoV-2 and this virus that causes COVID-19, it turns out that we actually knew or we know its sequence for quite some time. In fact, very early, starting in February, we already had information on its sequence. This is a figure from a paper that was published in April in Cell, so you can think of when it was submitted. And already back then, we knew that there were a few proteins that are part of this virus that are critical to the virus entry into the cell. And if you heard anything about the molecular biology of the virus, you should have heard about these proteins, ACE2 and TMPRSS2. It turns out that these are not surprising proteins because they were also identified as key entry proteins for SARS itself, SARS-CoV, which was the cause of the SARS epidemic. So this is something we have known for a while, but still there is a lot of research about the way the virus enters the cell. But a question that we haven't known or we couldn't answer back then, and we are still only starting to be able to answer, is what happens once the virus enters the cell. So it enters the cell through these two receptors, two human receptor, two proteins that bind to human receptors, ACE2 and this TMPRSS. But no, once it enters the cell, it actually activates a lot of other proteins, both viral proteins, and more importantly, it interacts with a lot of human proteins in order to activate whatever it needs to do. So the virus has a very limited capability in terms of the things it can do. Specifically, the virus cannot replicate itself. It needs the human machinery in order to replicate itself. In addition, the human machinery or the human immune response system is activating some programs to fight the virus. So a lot of things are happening after the virus enters the cell. And a question that we were very interested in, and a lot of people are interested in, is like, what exactly are these pathways? What exactly are these networks that are activated after the virus enters the cell? And this is very important because if we know what they are, and specifically if you can identify weak points in these processes, we may be able to develop drugs. So this is not about vaccines, which basically are ways for the immune system to learn how to recognize the virus. But even if you have not been vaccinated, if I know how the virus operates, I may be able to stop it. So this is the question that people have been looking at, again, starting in February, and basically employing a lot of new experimental methods that have been developed over the last 2 decades in order to characterize the activity of the virus. For example, one of the very important efforts was to determine what are the virus proteins interacting with when they are inside the cell, or specifically, the virus, the SARS-CoV-2 have on the order of 30 proteins that are encoded in its RNA. It's actually a pretty long virus. And it turns out that these proteins interact with human proteins. So they are not doing a lot of the work on their own because, as I said, the virus has limited capability, but it can hijack or activate some human processes inside our cells in order to perform its activity, to replicate, and so forth. So people have performed experiments. This is the paper that was published a few months ago from Nevan Krogan's group in UCSF. There have been other methods, other experimental attempts to characterize the set of proteins that interact with the virus. So this gives us some information on what happens once the virus enters the cell. So we know that once it enters the cell, it can interact with a bunch of proteins, not all the proteins, but a pretty large subset. It still doesn't give us the full picture of how these interactions are happening, what is the temporal process that are happening, what are the pathways and networks. So for that, people have looked at other types of data. For example, people looked at gene expression data. Gene expression data, or in this case, RNA-seq, looks at genes that are going up or down after infection in this case. So this can be time, or this can be different levels of the virus. And this specific example is a profiling of humans. So people that are actually infected with the virus, taking their blood and measuring the levels of genes in the cells. And the idea is that those infected, and then you have, of course, controls. And the idea is that genes that are going up or down following infections are likely involved in some either response or a pathway that the virus is using for its own benefit. So this gives us another view of what's going on in the cell, not in terms of the interactions, but in terms of levels. There are other ways to look at what's going on. There's something called phosphorylation that looks at which proteins change their shape once the virus comes into the cell, and a few other technological ways to look at what happens inside cells. So all of this information, all of this data has been collected, and a question that now becomes a major issue is, how do we combine all of these different types of data in order to get the full picture of what's going on? So some proteins interact with the virus, some go up, some go down, some change the structure. What is the entire process that happens once the virus enters the cell? And to do that, we need to actually use machine learning methods. And specifically, we need to use methods that can integrate different types of data and still represent a common network. And one of the major ways of doing this is to use a language, a set of machine learning tools that are called graphical models. So graphical models are ways to represent joint probability distributions. And if you have different types of data and you can provide some probability assumptions and relate them, you may be able to combine them in a joint probability space. So what I'm going to talk about next is some of the methods we developed for representing these joint probabilities and for integrating different types of data. And then I will talk about how we apply them to this specific problem of reconstructing the virus response pathways. So we have been working on this problem for several years, even more, I guess, more than a decade. And as I said, the main problem, or one of the main problems, is really how to combine all of these different types of data. So we have data about, this is protein-protein interaction. We have data about levels. We have other types of data, something called epigenomic data, which looks at how the DNA itself changes. We have information encoded in the genome, in the sequence, and so forth. Some of this data is static. So for example, the DNA, our DNA doesn't change during the infection. Once you get infected, your DNA doesn't change. It stays the same, but still it contains a lot of information that is used. On the other hand, some of the data is dynamic. The levels of the genes in your cells can change over time following infection and so forth. Similarly, the structure of the DNA can change and so forth. So some of the data is static, some are dynamic. And the question is, how do we combine all of these data sets together in order to get a clear picture or a unified picture of the activity and the networks that are activated in the cell? So one of the methods we developed for this is an extension of a very famous machine learning or graphical models method called hidden Markov models. And in hidden Markov models, the basic idea, for those who are less familiar with it, is that we have a set of states that are unobserved. In our case, the unobserved are the interactions that we usually are harder for us to measure over time. And we have a set of things that are observed. In our case, we assume that the observation, for example, are the levels of genes. And then you have, so this is the standard HMM, where you have states that control the emission, and you can transition from one state to the other. For example, infection at time point 1, infection at time point 2, and so forth. And in general HMMs, that's the structure, but we add to the HMM in this case something called an input layer. An input layer is a set of information that is static. It doesn't change over time, unlike the states or the emissions, but it can control or have an impact on the transition probability. What are these static information source? What is this input layer? We use it to encode the static information. So for example, in our case, the interactions that we measure, the protein-protein interaction between the virus and the host proteins, even though they change over time, are very hard to measure over time. So the information we have is only static, or information about which proteins regulate which genes. So this is encoded in this static layer. The levels of the genes is encoded in this layer, and this one encodes some state. And then we can use this to do inference and determine which interactions are driving states to the next state. So which interactions are regulating transitions, which in turn regulate emission, regulate the expression. So we use this to do inference on which interactions take place at which time point, even though we cannot really measure these interactions. And this is something we have applied widely to a lot of different biological systems, including to study immune response, but also development and others. And we're able to reconstruct a number of pathways and networks that are activated inside cells after some stimulation or after some development. one question that you need to address when you want to use these methods is, of course, how to sample. I'm sidestepping here because I want to then link it to the full model that we are developing. So, of course, there is this issue of how do you sample in time series data. And if you look at different studies that people have performed, right, because we want to know how these level changes, this is really critical. If you don't sample at the right time points, then even if changes happen between 2 time points, you will miss them. If you oversample, you waste money and time, so you don't want to do it. And if you look at different studies, even studies that did the same thing, this is just some data I collected on lungs, which are the key organ that is affected by the virus, you see that everybody is using very different time points. This is just a collection of time points for lung development. So over the last few years, we developed methods that can be applied to determine the optimal sampling rates for time series data. I won't go into the details, but I'll say that this is something we would apply as well when we study these networks in order to make sure that we don't miss any critical activity because of issues related to sampling. Okay, so this was generally the way we deal with static and time series data and how we combine it using graphical models, specifically with something called input-output hidden Markov models, and then how we apply it by determining the sampling rates and then sampling the expression level, combining it with the static information, with the static interaction data, and coming up with models. So all of this works reasonably well when the data related to the time series is really data that comes in various time points. What do I mean by that? If you can observe, let's say, a process that changes over time, for example, you know, you observe the sun at different times during the day, or I don't know, you observe the light outside at different times over the day, then you can say, look, this is the light I see in the morning, this is the light I see 4 hours later, 6 hours later, and so forth. And this is what we did in gene expression. So we infect cells with the virus at a certain time, and then we measure what happens 1 hour later, 2 hours later, 3 hours later, and so forth. But this relies on the fact that you have the same cells at each of the time points. If you take a cell out and measure it after 1 hour, and then you cannot use it anymore, there's no way for you to know what would happen after 2 hours and what would happen after 3 hours. So in the past, what we did is we, as a community, we looked at something called bulk changes. Bulk changes mean that we took a lot of cells, we would infect them, and then take a subset every hour and measure what happens and assume that whatever we see is some type of a representative response of the cell. That's fine, but it loses a lot of granularity because in the body and also in different organs, different cell types react differently. So individual cells can have different reaction, and when we aggregate them together, we may miss specific events in individual cells. So to overcome this, biologists developed technologies called single cell analysis, where you can actually take a single cell, which is a tiny object, and measure the levels of genes within that single cell. This is great because this provides information not on the aggregate, but really what happens inside each of the cells that this virus affects, but then it opens up new problems. For example, in this technology, once we take the cell out, unlike when we did it with the group of cells where we just randomly sample the subset, now when we take a cell out, we profile it, that's it, we kill the cell. So we don't really know what happens next. Right, obviously some cells in our mixture are very similar to this cell, and they would, you know, be in the next time point what this cell would have likely been in the next time point, but we don't know which ones are which. Right, so we have, let's say, 100 cells at time point 1. We take 10 out, and the other 90, some of them are related to cell 1, but we don't know which ones are which. So, you know, that's a major problem, and that's the problem of relating cells or trajectory inference in single cell data, right? So if we take a collection of single cells and we sample a few of them and we look at the rest, then we can say maybe, you know, these blue cells differentiated from the gray cells, and then green cells, and then these blue cells differentiated from these green cells, but maybe we are wrong. Maybe actually these blue cells came from the orange cells. We don't really know because once we take these orange cells out, we can't really know what happened to them next. So this is called trajectory inference. There's been a lot of work in single cell communities, but this is very important if we want to understand how the virus affects individual cells over time, we really want to have a technology that, first of all, can measure the differences, the changes in single cells, but also can tell us what the cell will look like at different time points. So linking these is another issue that we need to address if we want to build this big model. So for that, there's been several methods developed for that. We extended or further extended, I would say, HMMs to represent this sort of data. And now, unlike before, where we had these specific states, each state corresponded in our model to different times, so time point 1, time point 2, time point 3. In single cell data, the assumption is that cells, even though they are measured together at one time point, there is a big variation in their internal times. So cell can be continuously assigned to different locations. There is no reason to believe that all cells after 1 hour will behave exactly the same way. Some of them can be a bit early, some of them can be a bit late. If that's the case, then we need to be able to assign cells to continuous time points rather than to individual time points. Now, exactly how to do it, there are lots of ways. Some people have used dimensionality reduction methods to do it and then do some projection and look at paths on the low dimensional data. We didn't want to do it because we wanted to keep the original space. So what we did instead is we extended HMMs to have continuous states, basically to have infinitely many states. And then if that's the case, then you can actually associate a cell with any of the states or with any point along the trajectory, because the number of points is infinite, basically. Of course, when you do it in HMMs, you have to worry about some issues related to HMMs. For example, those who are familiar with HMMs know that HMMs have something called transition probability matrix, which is a matrix that defines the probability of transition in between different states. And when you work on a continuous HMM, you have infinitely many states. So you need to come up with an alternative way to define transition probabilities. But it turns out you can do it, and you can show that the definition, the new definition of the HMM or the continuous HMM is a valid definition for HMM, and you can still do inference and learning in this new model. In our case, we made it a bit more complicated because we had some constraints on which genes could change and so forth. So it wasn't a clean cut forward-backward algorithm, but you can actually do some a bit more or expand a bit the EM method in order to overcome that and really learn parameters for that. So we have basically continuous HMMs that can deal with the single cell data. And once you do that, you can combine them with the input-output HMM to identify not just the trajectory and where each cell lies, and I will show you an example in a second, hopefully this will be clear, but also what are the transcription factors and what are the regulators of this. Okay, so why am I telling you all this story about single cells? I'm telling you this because this turned out to be a very important method for us, at least, in order to be able to generate samples that we can use to study a infection by SARS-CoV-2. So I told you already that people have generated gene expression data, but most of the gene expression data that was taken from humans was blood data, right? Because it's very hard to go every day to the lung of someone and take a sample from the lung. It's a very, not the best or the nicest process to put a tube down the lung and get the samples out. So it's very hard to get lung data from humans over time. Blood is very easy. So you can do time series blood, but of course, the virus we know is mainly affecting the lung. So we would like to have lung data over time. How can we have lung data over time? One way to do it, which is what most people did, is to look at mouse. So you can take mouse, which is a known good model, but mouse have a very different phenotypes when infected with the virus. So I'm not sure mouse are the best model for this. So what's an alternative approach that on the one hand uses human data and on the other hand can still use lung-related data? An alternative approach is to derive lungs in the lab rather than take it from individuals. How can we derive lungs in the lab? It turns out that there are ways now to differentiate stem cells into different organs. So stem cells are these cells that can be derived into any cell. And there are protocols that can take stem cells and differentiate them into different organs. Even better, you don't even need stem cells. So there's technology called iPSC, induced stem cells, in which you turn it on you take skin cells from an individual, you turn on 4 transcription factors, 4 proteins, and they become, these skin cells miraculously become stem cells. This was the Nobel Prize, this won the Nobel Prize several years ago, and it's a technology that a lot of people use. So if we have a way to turn stem cells into lung cells, then we can generate these lung cells and now infect them in the lab with the virus and see what happens. Now, this can be personalized, right? Because I can take skin cells from anybody, do it, and see what happens in their lungs rather than do it on general lungs. So this is a very useful technique, not just for virus study, but also for personalized medicine. The problem is that these protocols for generating lungs that we really want for the virus are not trivial to implement, and more importantly, they don't end up with enough cells. So the protocols, basically what they do is they take these skin cells, turn them into stem cells, and then do some manipulation. So add some chemical, remove some chemical, and so forth. At the end, you end up with a bunch of cells. Most of them are not lung cells, and a very, very small fraction are lung cells. That's usually not enough. So one question we ask is, before we do these studies where we infect them, can we improve the set of cells that become lung and then use them for the, or we will want to use them for infecting with the virus. So for that, that's where we applied all of the methodology that I talked about. The first thing we wanted to do is decide how do we sample. So we have this protocol that people have developed that takes these stem cells, does something after 3 days, 6 days, and then from day 17, it starts treating them with some chemical, and then withdraw this chemical and so forth. And we decided to focus, and again, as I said, this protocol works, it generates lung cells, but it doesn't generate enough lung cells. So what we wanted to do is see if there is any way for us to improve the protocol by understanding why some cells become lung cells and some don't. And if we understand that, maybe we can intervene at some stage and convince more cells to become lung cells. That would be good for us, because then we can use them to study the virus. So this is what we have done, and we focused on this day, 2 weeks, just because it's a bit more, 2.5 weeks, because there were some indications that this is the problematic time point. And then, of course, this is there is roughly 17 days here. So the question is, when should we sample? Should we do every day, every 2 days, every 3 days? Of course, I mean, the more the better, but the more expensive. So we performed this time series, this sampling selection, and we selected, we determined the number of points, and it turns out that with 6 points you can actually get accuracy that is very similar to taking almost all of the points. So 6 points are pretty or are enough actually to sample for this time in order to reconstruct almost everything that you need. Okay, so these are the 6 points we selected, day 15, 17, 21, 25, 29, and 31. And we took some cells out of this, and then we applied these methods that I mentioned to reconstruct the models, right? So we have this method, and we have this HMM-based approach that initially clusters, then generates an initial structure, and then does the forward-backward algorithm to decide where to assign cells to state and what are the parameters, iterates, and at the end of the day, you end up with this model here. This model is the model or basically models the protocol. And what you see, these branches here are the different cell types. As I said, most of the cells start okay, so this is good, but then they branch out. This branch here is the cells that we are interested in. These are lung cells. All the cells at the top are cells that are wasting. Basically, they become gut cells or something else that we don't want. And the big question for us was, can we say what happens here? This is the key point. If we know what happens here, what drives some cells to become lung cells and some cells to become gut cells, maybe we can intervene and convince more cells to become lung cells. So this is a schematic. This is really the model that we obtained. Every dot here is a cell. So there is roughly 20,000 cells here. Every dot here is a cell. Every path is basically representing a time or a step in the process. But of course, cells are assigned continuously. So you can see they are assigned in across all the paths. This is the path we are interested in. This is the lung cells. And all of the cells at the top are actually not lung cells. They become gut cells and so forth. And the fractions of cells that end up here is pretty small. It's around 20%. So that's not good. And the question, again, we asked is whether we can intervene here and send more of these cells to become here. just to show that so of course, one question you can ask is, how do you know that intervening here would help? Maybe, yeah, you see the split here, but maybe this was already predetermined way earlier. You just don't see it in the expression, but there is something going on inside the cells that makes them, they have to go this way and these have to go this way. So there are actually ways to show it. I won't go into the details, but you can actually show that that's not the case. There is still plasticity, there is still ability for cells to become lung cells at this point. They have not been committed to the cell type earlier. Okay, so that's something we can show. And if that's the case, then let's see what's the difference between the ones that go up and the ones that didn't. And we, again, this is some biological exploration, but using the model and some knowledge from our collaborators, we narrowed it down to something called the Wnt pathway. Wnt pathway is an important set of genes that are working together in order to activate some signaling. And if you look at components or members of this pathway, you see that they start to diverge. So if you look at the path of the cells, they start to diverge after some time. Cells that we are interested in have this repressed. So basically this is not activated, and cells that become gut cells are actually activated. And this is true for a number of these. So this maybe indicates that if we can suppress this pathway at this time, we can send more cells in the right direction. So the question is, what can we do? And second, what is this time? So this is the time for the model. We need to translate that into real time, and that's something that we can do using the model. So we decided that this time is day 17.465. We allowed them to round it a bit to 17 and a half, but if you want to really be good, 17.465. And then the idea is that at this day, we want to intervene and basically reduce, in this case, remove the Wnt signaling or chemical that leads to Wnt signaling. And when you do that, you actually gain almost twice as many cells as you would get in. So this is the control. This is like 25% that you get if you just apply the regular protocol. But if you apply the protocol that removes this chemical that activates the Wnt signaling pathway at day 17, as predicted by the model, you get roughly 45% of the cells. You can remove it later and still get improvements, but the improvements are much weaker. And in fact, at some stage, it doesn't help at all. So if you remove it at day 19, it helps a bit. If you remove day 21, it doesn't help anymore. So this means that, first of all, the removal helps, but also the temporal prediction is important in this case. It's not just saying, knock out this protein or remove this pathway, but you also have to say when in order to get the cells. Okay, so this was some detour, but after this detour, basically, we have a way of generating lung cells in the lab, and now we want to know how can we use them in order to understand how the virus infects the cell. So that's what our collaborators did, and they recently published a paper in which they profiled basically so basically, they took the protocol that I just described, this improved protocol. They generated the lung cells, type 2, right, lung cells. They infected them with the virus, and then they looked after 1 day, so control 1 day and 4 day. There are other data sets. So this is one data set we looked at. There are other data sets. So this is basically gene expression following virus infection. And as I said, we wanted to combine it with others. Okay, so far we looked primarily at what happens inside the cell in terms of the regulation of genes, which genes go up and down. But when virus infects cells, there's more things going on than just the expression or activation of genes. For example, when a virus enters the cell, in this case, let's say it enters through these 2 receptors, it goes in the cell, right? It goes in the cell, and inside the cell, it activates some type of a response that we can observe using gene expression, like I've shown you now. But that's the gene expression. The virus is the input, the gene expression is the output, but it's not directly linked, right? There's things that are going on inside the cell that link the two. One of them is the transcription factor or the human proteins that activate the gene expression that respond to the virus. And the second is this protein-protein or virus-protein interaction that activates the human proteins that in turn are activating the response. So the question is, can we use the methods that I just described to infer not just what happens in the transcriptional response, but also in the signaling or the virus host interaction response? So for this, we combined a number of different data sets. So one is time series data. As I mentioned before, we already have a model for that. But now we also want to use protein interaction. So human proteins that interact with the virus, and this is something conditioned. This is specifically for the virus. And we also have general information about protein DNA interaction, motif information, and protein. And this is the type of methods, or this is the type of models we would like to find. We want to find cases where we can see a viral protein interacting with a human protein. And at the end, at the end of this chain, the human proteins activate a transcription factor, another human protein, which in turn turns on the genes, and that's what we see. So if we can reconstruct this process going from the viral protein through the signaling network to the human response, we basically reconstructed what happens inside the cell. There are some computational issues involved in reconstructing this. It's not a trivial thing, even if you know the interactions, right? So I said we can measure human virus host interactions. We can measure human protein. There's a lot of databases. But unfortunately, the hypothesis space is very large. So you really need to constrain the hypothesis space, because otherwise you get too many paths going from viruses to the transcription factors that we're interested in. So we looked at a number of different ways to constrain these hypothesis spaces in order to really narrow down and figure out what are the most important pathways or networks. And for that, we came up with a few optimization methods. Some of them, for example, are asking to find the best, or so basically we're looking for orientation. So these protein-protein interactions are not oriented, they are undirected, and we want to direct them to get a pathway going from the virus to the host, to the transcription factor. And one thing we can say is, give me an orientation that maximizes the number of ways you can get from a viral protein to a human protein. This turns out to be an NP-complete problem or NP-hard problem, but you can find approximation for it using some stat solvers. You can also define this as an integer programming problem. I won't go into the models, but these are things we have worked on a few years ago, and you can find solutions. Again, even for this is integer programming, so you don't have an optimal solution, but you can have an approximation. Once you have that, or once you solve these approximation problems and find a way to orient the edges going from the virus to the transcription factors, basically you can reconstruct the set of interactions that lead from virus proteins to the proteins that activate the genes. And then we have a separate part that uses these proteins that activate the genes to infer what is the set of regulators and how are they affecting the genes at each time point, which is what I showed you. So we have this two-stage process. One is looking at what we call the signaling network, and one is looking at the regulatory network. But of course, there are links between them, because if you have transcription factors that activate the gene, you want them to be connected to the viruses, because somehow they were activated because of the virus response. On the other hand, if you have a virus response, you want to see, or if you have a virus interacting with a human, you want to see how this basically affects the gene expression. So what we really do is we do this iterative process where we learn this regulatory network, we see how well we can connect it to the virus proteins, and based on that, we iterate selecting proteins that activate genes based on how well we can connect them, until we end up with a model for that. And this is the model that we ended up with for the virus, for the SARS-CoV-2 interaction. Let me just explain what the network shows you, and then discuss what you can do with this network. So in red here, we have human proteins that interact directly with the virus. So we know from prior studies that these interact directly with the virus. In green are proteins that don't interact directly with the virus, but our method identify them as linking those that interact directly with the virus with those blue proteins that are called transcription factors, don't interact directly with the virus, at least most of them don't, and they are the ones activating the genes. So this is the process, if you think about information flow from the virus to the human response that later can activate or help or fight the virus itself. These are the proteins that directly interact with the virus proteins. These are the intermediate signaling molecules. And these are the transcription factors that activate the virus. Now, this is the network that we came up with. There's lots of ways to validate this network. How do you know that it's correct? One way to validate is to look at independent analysis about which proteins are actually phosphorylated. Phosphorylated means that they change something about their shape. following virus infection. And in this case, all the diamond proteins are phosphorylated. You can see that a lot of the green proteins are phosphorylated, even though they are not directly interacting with the virus. They are affected following, we know actually from additional studies, of course, the network identified them, but even without the network, we know that a lot of them are affected by the virus, or at least phosphorylated, basically activated following virus infection, and similarly some of the direct targets of the virus. So this is this is one part of the network. This is the signaling, and this is the second part of the network. The second part of the network are these transcription factors that activate the gene expression that we saw. So remember, we started with this gene expression response. This is the first thing we used, and in order to find what drives directly this gene expression, these are the transcription factors we identified. So this is the network leading to the transcription factors, and all of the proteins listed here are transcription factors, and they're also listed here as regulating different responses. And here I'm plotting the response of genes. These are genes that go up at the first time point, and some of them go up next. And these are transcription factors that regulate them. And again, we are highlighting some of them that are themselves expressed. A lot is known about these transcription factors. For example, these are cell cycle regulators, these are immune response regulators, and so on. Okay, so this basically provides a network or a view of the activity of different networks in the cell, signaling and regulatory networks following the infection with the virus. We next wanted to prioritize these proteins that we have here and here in order to find those that we should highlight for testing about whether we should target them with a drug, whether if we remove them, it can impact, or if we overexpress them, activate them, or remove them, it can impact the ability of the virus to actually replicate itself or to increase its load in the cell. How can we do that? We do that by intersecting the list that we found with other information. And what other information can be useful? For example, we can look at information about underlying conditions. So we know that a mortality and even infection from SARS-CoV-2 is associated with a lot of other underlying conditions. For example, hypertension, heart disease, diabetes, obesity, and so forth. If that's the case, it means that for these individuals, they express or repress a set of genes that the virus really likes or really likes to be repressed. Otherwise, it's unlikely the virus works so well on those and it doesn't work well on others, right? So it turns out that if you look at genes that are overexpressed or underexpressed in these conditions, you may actually be able to find genes that are responsible for the virus activity. Now, if we intersect the set of genes that are activated in this condition with the set of genes that we found, we can find, or we can start to zoom in on these genes that the virus really needs in order to be very efficient in replicating itself and increasing its load. So that's something that we did. And if you look at these genes, we found a subset of the genes, and this is a small subset on the order of 30 of the hundreds of genes that we found that are on the one hand identified by our network, but also identified in several of the underlying conditions. So these are the conditions, these are the set of genes, and this allows you to narrow it down. And if you look at the biological processes that these genes are involved with, you can see that a lot of them are known to be response to stress, viral response, so these make sense. So at the end of the day, what we got is this combination or intersection of network identified proteins that we can rank. I won't go into the details, but there are ways for us to rank them. And we also have a set of genes that we identified based on underlying conditions. And the intersection is 45 genes. Now, if you look at these genes, you can ask, are these genes really impacting virus loads? How can we say? We can do some experiments. And it turns out people have done experiments. Specifically, people have done something called CRISPR knockout experiments, where you remove certain genes and you see their impact on the activity of the virus. Now, of course, they were not done in the cells that we looked at. They were not done in the condition that we studied. So there's a lot of differences between these experiments and what we've done. But this is at least a good indication, or at least a possible way to look initially of whether the predictions that we made are correct. And here I list a number of proteins that were identified by other studies that looked at knockout and were also identified in the 45 lists that we found. And many of them are not sources. So many of them don't directly interact with the virus. So the only way to identify them is if you reconstruct the activity in the cell. If you just look at those that interact with the virus, you miss most of the genes on this list, although a lot of them are known to affect the virus. So this is SARS. More importantly, or maybe more generally, we actually have a number of genes identified by our model in different gene expressions. So the BU collaborators or single cell expression, or in both of them. And a lot of them don't interact directly with the virus, but several of them actually have approved drugs. And if you have approved drugs, it's much easier for us to test because we can just grow the cells in the lab, apply the drug and see if it helps. And if the drug is approved, it should be the target, the gene should be the target of this drug. So this is the next step for us. Our goal is really to take these cells, grow them in the lab, infect them, treat them with the drug that we identified for this specific protein, and see the impact on the activity of the virus in these cells. Okay, so this is where we are at. Basically, we were able to reconstruct some of the networks using gene expression based on protocols we developed for generating lung cells from stem cells. We infected them with the virus. We identified significant expression changes, and then we integrated that with a lot of other data, not from our group, but from other groups, and came up with models for the virus. Now, just to conclude, I think, and this is something that I think a lot of people believe, is that in order to understand what's going on inside the cells, we really need to integrate a lot of data sets. So it's important to measure the activity of the virus, the expression, the interactions, the phosphorylation, the protein DNA and so forth. But really the challenge is to put all of these things together. And then once you put all of these things and you have networks, it's not enough. You will still have a lot of possible hypotheses. You need to further filter that out. And one way to do it is to look at genes that are known to be relevant to some underlying conditions. And then once these are identified, of course, the next step is to test them and see whether or not any of them can can basically lead to or can impact the virus itself and the virus loads. And as I said, our goal is to test it on the ones that have approved drugs. Another component of our project is to develop new drugs for proteins that we think are very important, but the drugs do not exist. So we have another collaborator from MIT who is developing deep neural network methods to identify compounds, small molecules that can impact specific proteins, and then use those to treat the proteins that we think are very important, but for which no drug is currently known. So that's it for us and where we are at. As I said, next step, or really our goal is really to test some of these predictions, at least in terms of the ability to knock them out. So this is our group, and of course our funding from C3, the Digital Transformation Institute, as was mentioned. We are funded, or we are one of the awardees in this program, but we are also funded by the NIH. This is my group at good time, so the last gathering, and this is the group, recently following social distancing, where only a few can attend, but hopefully we'll go back after we are able to find good drugs for this virus. We will go back to the bigger meetings. And that's it, and I'm happy to take questions.
Thank you very much for an excellent presentation. And I have to say, this is Costas again. I did go to CMU a long time ago. I did recognize the location of your picnic, which is nice to see. So do you have any questions before I turn it to, I have a question myself, but before that, do you have any questions from the other panelists before we start? If not, I can get us started. I was looking at your proposal, and there was a phrase there that kind of, stood out. You want to develop prediction algorithms that can robustly extrapolate beyond their training data. Okay, extrapolating beyond the training data. And I understand this is a general question. I wonder, how does this play in your goals, and how do you confirm that this extrapolation is actually robust and correct?
Right. So if I don't remember exactly the words that we used. I can, if I had to guess, I would say this is related to the part of trying to predict drugs or small molecules for specific proteins. And there the goal is really to look at the structure of the proteins and look at the chemical composition of the compounds and try to be able to learn deep neural networks that link or that try to infer features from the proteins in order to identify what are the drugs or what are the molecules that can treat these proteins. But more generally, I think in biology specifically, and this is maybe a unique case in machine learning, but if you work on biology, then of course you can do a lot of train tests. As I said, for example, in our case, right, we have these set of proteins that we predict are very important for the virus. People have done experiments in the past where they do knockout or something called knockdown. They remove the gene and they can test the impact of that knockout on the cell. So that's definitely something you can test with current data that is available, but that's not enough. The real challenge, I think, for a computational modeling or a computational systems biology approach is to drive new experiments and to make sure that these experiments can validate your results. So just doing train, test, and building a model that works very well for the test data is nice. Usually biologists don't appreciate it because it doesn't tell you anything new. We knew that these are the validation. We knew that this is the training data. Now tell me what is new and show me that it's really working. So one of the nice things I think about biological analysis, especially molecular, is that if you have hypotheses, and in our case we do have, then you can really test them in the lab and then find new proteins that people have not shown before, but you can show based on your model that are really affecting, in this case, virus loads, but you can think of other cases.
So in other words, there is empirical data in the field that might be used to show that what you have extrapolated indeed has some basis.
Well, so there is empirical, but I think that's less exciting. I think the exciting thing is the data that doesn't exist yet, and you can generate after you have the model, right?
I see.
So those 20,000, I mean, those 20,000 human proteins, even more, but let's say 20,000, right? There's no way you can test all of them in all of the configurations, in all of their conditions. So if you have a good model that works on whatever is in the field already, I think the exciting thing is to say, okay, this is the prediction and let's do new experiments and find out new biology based on that. And that never existed, but based on your model or your method, you can find that out. And I think that's what's exciting to a lot of biologists, including, and I agree. I mean, I think that's, for me, that's something that's very exciting in this area.
Very good. Do you have any other questions from the panelists or from the attendees? Of course, I have another general question myself, admitting not being in the area. I'm actually kind of intrigued. Could you walk us in a very simple way on the steps that it will take after the work on the lab is done towards the actual production of drugs for human use? What are the steps in general and how long would it take?
Yeah, well, yeah. So if you're, I'm also not in this area. I can tell you. So one of the exciting things, or not new, but something that people have been excited about for 10 years or 15 years now, is actually something called drug repurposing. Drug repurposing means that you take a drug that is already approved and you find new uses for that drug. That's actually pretty easy to get. I mean, not trivial, but it's it's possible to get that to being administered, because the drug, if the drug is approved, it means that it's passed already very serious safety protocol and evaluation. So the drug is safe. Usually the drug that is approved is safe. Whether or not it works for this new condition, usually these are conditions that no drug, I mean, if another drug works, they will use it. But if nothing works, then the worst that could happen is that you're not going to make things better, but you're not going to cause any harm because you know that this drug is already approved. So drug repurposing has emerged as the major goal of molecular and systems biology, because this is taking a drug that we already know works and applying it to new things that we didn't know. So if you have a drug that is approved, it's reasonably, the steps to get it approved, especially for, I mean, by the way, for SARS-CoV, for SARS, it's actually much easier because not a lot is known. So it's pretty easy to get at least the clinical trial. If you come up now with a molecule that nobody tested before, and you know, and you want to approve, and you claim that it works very well for a specific protein, and that once you are once you remove that, or you apply the drug, the protein is removed, and the virus disappears, that's extremely, that's much more challenging, because first of all, you have to show that it's true, but even if it's true, maybe it does a lot of other bad things that are much worse than treating the virus, right? Because the virus maybe affects 2%, or maybe is deadly to 2% of the population, but if you do something else, if you cause cancer, it can kill 10%, so you haven't done anything. So getting something to the safety level is very hard. That's why, for example, vaccines, right? People are now talking about vaccines very quickly. The problem with vaccines and with drugs is that vaccine you apply to everybody. So it's enough that the vaccine is causing, I don't know, 0.1% harm, that you have to be very careful. That's why people are extremely careful with vaccines, and similar with drugs. So new drugs or new vaccines are very hard to get into. Drug repurposing is much easier.
Thank you. Now, there are a couple of questions on the Q&A panel. Maybe you can read them directly. I want to make sure that...
Sure. Right. So the first question was about directed versus indirected. That's true. So most of the protein, in fact, all of the protein interaction data is undirected. Protein DNA interaction is by definition directed, because protein regulates DNA. So one of the things both in the IP, but more importantly in the satisfiability problem, we are trying to find the correct directions that we will use in order to find the paths from the virus to the transcription factors. Now, if you think about it, the main challenge is that we don't allow bidirectionality. If we allow always one protein to be directed to another and the other, then there's no problem. But if we insist on one direction, then there are some paths that you can take, but some paths you cannot take. And you can show that if you want to maximize the number of paths that satisfy some connectivity, this is an NP-hard problem. So you need some approximation algorithm, or you need some IP solvers in order to do that. So that's for the directionality. And again, that's a very interesting problem. I think generally speaking, in protein interaction, not just for virus, but also to infer other types of signaling networks. And people have worked in this in the computational biology area. So that, yeah, so directionality is important. Yeah, so in terms of the continuous, so the second question was about continuous HMM and the fact that I claimed that there's infinitely many states. So what do I mean by where is the state, or what is the state mean? So in this case, yes. So the continuity, or the state is for a cell. A cell is also an observation. So each cell has each cell is an observation. We do have a time associated with that observation, but we don't believe that this is the real time. So for example, I can measure a cell after a day, but it could really be that this cell is more similar to cells that I would measure after a day and a half, or a cell that I would measure after a half a day. So some cells are a little bit farther along, some cells are a little bit backward. So we want to associate a specific time or a specific state, which represents in our model time, with each of the cells. So each cell has an observation. Now, a state can have multiple cells. Of course, if we're talking about continuous, then it's almost impossible that the two cells will have the same state. So each cell has a specific time associated with it. In practice, because we are doing inference, we actually sample a number of states. So usually we sample 100 states, maybe more, depending on the model, although the model can be used to sample as many states. So we can eventually have multiple cells assigned to the same state, even though it's continuous, because of the way we do inference. But in practice, you can sample as many as you want, and if you sample a lot, then it's unlikely that two cells will have the same state.
Thank you. Do you have any other questions? If not, I see that we are actually out of time. So thank you very much for an excellent presentation.
Thank you. Thank you for inviting me, and thank you for supporting us. We are excited about this project.
You're welcome. And I hope to see you all next week, same time.